Hello everyone. Today we are going to be going through the process of photosynthesis and how it differs between C3, C4 and our CAM plants. Now, photosynthesis is simply the process by which plants turn light energy into chemical energy. This form of chemical energy is known as glucose in this case. Glucose is a 6-carbon molecule and it also has 12 hydrogens and 6 oxygens. Glucose is a sugar, which can then be used as an input into cellular respiration to produce a more usable form of energy that we call ATP. So this is the most usable form of energy used in all cells. However, I think this cellular respiration discussion will be a video for another time. Right now, let's focus on the process by which we turn this light energy into glucose first. So plants turn this light energy from sunlight into their food, a sugar. This food can then be input into cellular respiration to produce a more usable form of energy called ATP. So photosynthesis is essentially the process through which plants create their own food, as plants are autotrophs, meaning that they create their own food, they create their own glucose. Contrastingly, humans get this glucose through the food that we consume, whereas plants, they create their own food. Now, before we delve into the details of photosynthesis, it is important to understand the structure of the chloroplast. The chloroplast is an organelle found in our plant cells. It is composed of 2 membranes, so 2 phospholipid bilayers. So it's got an outer membrane and an inner membrane. These chloroplasts also contain their own ribosomes, which are different to what we find within the rest of our cell. So the ribosomes found in our cytosol are different to the ribosomes found in our chloroplasts. These ribosomes are smaller than the ones that we find in our cytosol. This is because chloroplasts are thought to have originated from a free-living photosynthetic bacteria. It is thought that approximately 2 billion years ago, these free-living photosynthetic bacteria were engulfed by a larger host cell. Therefore, it makes sense that these ribosomes are more similar in structure to bacterial ribosomes. So they're similar to bacterial ribosomes rather than our eukaryotic ribosomes found within the cytosol. Because of this, we also find that the chloroplasts have their own DNA, their own circular DNA. And this circular DNA, just like in bacteria, is not enclosed within a nuclear membrane, like our eukaryotic DNA found within the plant cell's nucleus. So this DNA is circular, whereas the DNA that we find within our plant cell's nucleus is linear. This DNA is still double-stranded, but is in a circular form instead. And now, most importantly, we find these membranous structures within our chloroplasts organized in stacks, which we call a granum. So this stack is called a granum. Each component of this stack, so one of these oval shapes, are known as the thylakoid. And our very first step of photosynthesis occurs within our thylakoid membranes. So our thylakoid membranes are very important because this is where our light-dependent stage of photosynthesis occurs. Last but not least, this fluid-filled space on the outside of the thylakoid membranes, but within our chloroplast, is known as the stroma. So this is known as the stroma. And this structure is also quite important since this is the location for our second stage of photosynthesis, known as the light-independent stage. Alrighty, now that we understand the structure of our chloroplasts and we have learnt the locations of our light-dependent and light-independent stages of photosynthesis, let's move on to discussing the purpose of each stage. Now, the following equation is often used to represent photosynthesis and represent its inputs and outputs, so its net inputs and outputs. As we discussed before, we require light energy, so light energy, for this reaction to occur. In order for us to harness or capture this light energy, we also need a pigment known as chlorophyll. This chlorophyll is found within the membranes of our thylakoid. Now, if we've ever owned a plant before, I think we'd all be aware that plants need water in order to survive. We also know that plants release oxygen into our atmosphere, and this is the same oxygen that we require to breathe. This oxygen is only able to be released in the atmosphere due to the presence of water. A very important step, which we will be going through, is the splitting of this water molecule in the light-dependent stage. The splitting of this water molecule into its hydrogen and oxygen components allows for the release of oxygen into our atmosphere. Another reason why plants are so important is because they absorb carbon dioxide in order to make our glucose molecule. As we can see here, this is our only source of carbon. And this carbon, these 6 carbons, are used to generate this 6-carbon sugar known as glucose. Now this equation, though it gives us an idea of the source of our carbons and the source of our oxygens, this equation is actually simplified. And it is simplified because we don't actually have 6 molecules of water input for 1 molecule of glucose to be produced. We actually have 12 molecules of H2O input, and we have 6 molecules of H2O generated in the light-independent stage of photosynthesis. When we simplify this equation, we minus 6 H2O from both sides, leaving behind none on the product side and 6 on our reactants side. So, if you ever see the photosynthesis equation written like this, know that this is simply the unsimplified form of photosynthesis. If we write it the simplified way, we should only have 6 H2O in our reactant side and no water molecules in our product side. However, once we go through the process, you will see that though we need 12 molecules of water input into this process, we also generate 6 molecules of water in our light-independent stage as well. So our net use of water is 6 H2O molecules. Alrighty. As mentioned before, photosynthesis can be split into 2 stages. First, we have our light-dependent stage, and then we have our light-independent stage. The purpose of our light-dependent stage in our thylakoid membranes is to produce high-energy molecules. And these high-energy molecules are known as ATP and NADPH. Now, it is important to understand that ATP and NADPH are produced due to reactions that occur within the cell. ATP stands for adenosine triphosphate. This means that this molecule has 3 phosphate groups attached to it. For ATP to be produced, we use its low-energy counterpart known as ADP, which stands for adenosine diphosphate. This means that the ADP molecule only has 2 phosphate groups attached to it, and this is the reason why it is known to be a lower energy molecule than ATP. So, in order for us to produce ATP, we require a phosphate group to be added to our ADP molecule. So we require a phosphate to be added to our low-energy ADP molecule to create our high-energy ATP molecule. Our NADPH, however, is produced from a reaction that adds hydrogen molecules or protons to NADP+, which is known as the low-energy counterpart of NADPH. So just like ADP, our NADP+ is known to be the low-energy counterpart of NADPH. When we add a hydrogen to it, it becomes the high-energy molecule known as NADPH. However, all of our chemistry students over here, we should know that this chemical equation is not yet balanced. Our charges on either side are not yet balanced. We have 2 positive charges on this side, but a neutral charge on the product side. This means that we must neutralize these 2 positive charges by adding 2 negative charges, known as electrons. So this is actually the equation for how our high-energy molecule NADPH is produced. So we take our NADP+ molecule, we add a hydrogen and 2 electrons to produce our high-energy NADPH. The purpose of our light-independent stage is therefore to create glucose using the high-energy molecules that we made in the light-dependent phase before it. So the purpose of this one is to create glucose by using the ATP and NADPH produced in the previous step of photosynthesis. So the ATP and NADPH produced in the light-dependent stage of photosynthesis. This means that in the light-independent stage, we have the opposite reaction occurring, meaning that our high-energy molecules will be converted to their low-energy counterparts. So our ATP molecules will be converted to ADP and a phosphate group, and our NADPH will be converted to its low-energy counterpart, NADP+, a hydrogen ion, and 2 electrons. Now, although VCAA does not require you to know the exact detailed mechanisms behind the light-dependent and light-independent stages, roughly understanding the overview of these processes will be helpful in memorizing the inputs and the outputs of each of these stages. It will also assist in understanding why our oxygen here comes from H2O and not carbon dioxide. So, let's delve into the light-dependent stage first. Now, we already defined that our light-dependent stage of photosynthesis actually occurs within these thylakoid membranes. Just like any other membrane, our thylakoid membranes have a phospholipid bilayer. So here's our phospholipid bilayer of our thylakoid membrane. Again, like all membranes, the thylakoid membrane actually has proteins embedded within it. Though you are not required to know the names of these proteins, it is helpful in understanding how and why ATP is able to be made within this space. As mentioned before, our chlorophyll molecules are actually found within particular proteins on this thylakoid membrane. The role of this chlorophyll is to absorb light energy and allow for the excitation of electrons. The absorption of this light energy also allows for the very important step of splitting our water molecules. So our water molecules are split into their hydrogen, so two hydrogens, and their oxygen counterparts. If we balance this equation and remove any fractions here, we will know that for every 2 H2O, we get the production of 4 H pluses and 1 O2 molecule. So just 1 O2 molecule. So over here, 2 times 2 for the hydrogens gives us 4, and 2 times 1 oxygen will give us 2 oxygens here, which are released in the form of O2. So this is the form of oxygen that we breathe. Now again, we should know that this equation is not yet balanced in its charges, meaning that this side is neutral, our reactant side is neutral, and our product side at the moment carries a charge of +4. So this +4 charge must be neutralized by 4 electrons on this side. So this means that in the splitting of 2 water molecules, we get the production of oxygen and 4 electrons over here. These electrons can eventually be found within these proteins and within our chlorophyll, and so when light hits the chlorophyll, these electrons can go into an excited state. This excited state of the electrons allows for it to do 2 things. So our excited electrons travel through certain proteins on the membrane and eventually reach a protein known as NADP plus reductase. This NADP plus reductase is an enzyme on the membrane which is responsible for catalyzing the reaction of NADP plus to NADPH, which we discussed before. So the electrons that we get over here are used within this reaction, which we know is NADP plus a hydrogen and 2 electrons. So these 2 electrons would have previously traveled through all of these proteins until it got to NADP plus reductase in order to produce some NADPH. Now we know over here that 2 electrons are required to produce 1 NADPH, and we also know that 2 water molecules release 4 electrons. This means that with 4 electrons, we should be able to produce 2 molecules of NADPH, as only 2 electrons are required to make 1 NADPH. So 2 electrons times 2 will give us 4 electrons. So the 4 electrons over here can make NADPH twice. Another role of these excited electrons is that as they travel through these proteins on the membrane, they allow for more hydrogen ions or more protons to be pumped into this internal space of the thylakoids. So we get more hydrogen ions floating around in this space. This is why the internal environment of our thylakoids is known to be acidic. So anywhere that we have lots of H pluses, lots of hydrogens, we know that this area will be acidic. We also get protons or some H pluses from the splitting of water, but we also get H pluses pumped into this space via the action of these excited electrons traveling down the proteins located on the membrane. This gradient of hydrogen ions is very important because the inside of the thylakoid has a high concentration of H+, but the outside, so the stroma area, has a low concentration of H+. This is important because we have another very important protein located within our thylakoid membranes, and this protein is known as ATP synthase. Our ATP synthase is responsible for catalyzing the reaction of ADP, so turning ADP into ATP. So it is responsible for catalyzing the addition of the phosphate group onto ADP to create the high energy molecule ATP. However, this reaction, or this ATP synthase, can only catalyze this reaction as long as there are hydrogens flowing down their concentration gradients from the inside of the thylakoid to the outside of the thylakoid. So as long as we have this concentration gradient, our hydrogen ions can flow from a high concentration inside the thylakoid to a low concentration outside the thylakoid through the ATP synthase. And the ATP synthase can then harness this energy, this kinetic energy produced by the flow of hydrogen ions, to catalyze the production of ATP. So if we summarize the inputs and outputs of our light-dependent stage, we know that water would be our most obvious input. We also know that the purpose of our light-dependent stage is to produce high-energy molecules such as NADPH and ATP. However, for NADPH and ATP to be produced, we require their reactants. So for NADPH to be produced, we require input of NADP+, and for the production of ATP, we require the input of ADP and a phosphate group. We also know that from the splitting of this water molecule, we get production of O2. Now let's try to quantify how many of each of these inputs and outputs are actually required. Now we already know from our photosynthesis equation that 6 O2 molecules are produced. We also know that for 1 molecule of O2 to be produced, we require 2 molecules of H2O. So we must require twice the amount of water than the amount of oxygen. So it makes sense that we have 12 H2O as our input at this stage. So this is where the 12 H2O from our unsimplified photosynthesis equation comes from. So the 12 H2O that we discussed in the unsimplified equation is input into the light-dependent stage. Now we also know that for every 2 H2O, we get production of 4 electrons. We also know that for every 4 electrons, we can create 2 NADPH, because to create 1 NADPH, we only require 2 electrons. So with 4 electrons, we should be able to create 2 NADPH. So for every 2 H2O, we get 2 NADPH. So for every 2 H2O, we get 2 NADPH. So we have the same amount of H2O to NADPH over here. They are within the same ratio of 2 to 2 or 1 to 1, if simplified. So it makes sense that we would get production of 12 NADPH. So this will be 12 NADPH. Of course, to produce the 12 NADPH, we would require 12 NADP pluses for the hydrogens and electrons to be added to. Now, our ATP production actually depends on the activity of our ATP synthase. However, without understanding too much about the exact structure of ATP synthase and its mechanism of action, we can say that there are 12 ATP produced at this stage. Note that most biology textbooks state that for every 2 H2O splits, there are 2 to 3 ATP made by ATP synthase. According to this, we can have anywhere between 12 to 18 ATP made during one light-dependent stage. So any value between 12 to 18 here would be considered accurate. Alrighty, so this was a rough overview of our light-dependent stage. Let's move on now to our light-independent stage of photosynthesis. The light-independent stage is also sometimes known as the Calvin cycle. As the name suggests, this process is a cycle which allows for the production of glucose. In this cycle, the last product of our cycle, known as ribulose bisphosphate or ribulose 1,5-bisphosphate, is turned into a 6-carbon intermediate, which then splits into 2 3-carbon molecules, which undergo further transformations to once again produce ribulose bisphosphate. Our ribulose bisphosphate is actually a 5-carbon molecule. So this is a 5-carbon molecule. Now we know that the first step of this Calvin cycle requires the production of a 6-carbon intermediate. The way that we produce this 6-carbon intermediate is by adding carbon dioxide, which is one of our inputs of photosynthesis, to our ribulose bisphosphate. So the reaction of ribulose bisphosphate and carbon dioxide allows for the production of this 6-carbon intermediate molecule, which then splits into 2 3-carbon molecules. This addition, or this step, sometimes referred to as carbon fixation, is catalyzed by an enzyme known as rubisco. Rubisco is very important for this first step of carbon fixation to occur. This initiates our light-independent stage of photosynthesis and allows for the production of the 6-carbon intermediate and allows for the rest of the cycle to continue. In this cycle, these 3-carbon molecules are known as 3PG, or 3-phosphoglycerate. This 3PG molecule then undergoes transformations to once again produce ribulose bisphosphate. However, for all of these transformations to occur, we require the use of our high-energy molecules ATP and NADPH. So in this stage, our ATP, our high-energy molecule, gets used, so its energy gets used, and it is converted into its low-energy counterpart, ADP, and a phosphate group. Similarly, this NADPH, this high-energy molecule, is used and converted into NADP+, and hydrogen and the 2 electrons to balance out the equation. So after our initial step of carbon fixation, we have the steps of reduction and sugar production. This step of reduction and sugar production requires the use of 2 ATP per carbon dioxide molecule and 2 NADPH per carbon dioxide molecule. This second step allows for the production of a 3-carbon molecule, and 2 of these 3-carbon molecules can be combined to produce glucose. However, to get from this form of this 3-carbon molecule, so to get from the 3PG form of this molecule to a different form of this molecule, we require the use of 2 ATP and 2 NADPH. So these 3-carbon molecules, 2 of them can be combined to create 1 molecule of glucose. Our last few steps of the Calvin cycle are known to be for the regeneration of ribulose bisphosphate. For this regeneration to occur, we require the use of 1 ATP molecule per carbon dioxide used. For 1 molecule of glucose to be produced, we require this cycle to occur 6 times. This is because some of the 3-carbon molecules produced over here continue on through this cycle to regenerate ribulose bisphosphate. So in order for us to get 2 molecules of this 3-carbon molecule to come out of this cycle and produce glucose, we require 6 cycles from each of the 6 carbon dioxides that are input into photosynthesis. So once again, if we try to quantify these numbers, we will get this cycle occurring 6 times. So 6 carbon dioxides will be input here in total. We said before, for the reduction and sugar production stage, we require 2 ATP and 2 NADPH per carbon dioxide. So we require double the amount of ATP and NADPH compared to the number of carbon dioxide. So in total, we would require 6 times 2, so 12 ATP and 12 NADPH in the reduction and sugar production stage. For the regeneration of ribulose bisphosphate stage, we know that we require 1 ATP per carbon dioxide molecule. So we know that 6 times 1, we would require 6 molecules of ATP in this stage in total. Similarly, if we quantify the numbers of ribulose bisphosphate, we know that 6 ribulose bisphosphate will have a carbon dioxide added to them to produce 6 of these 6-carbon intermediates. These 6-carbon intermediates will each split. This means that we will have 12 of these 3-carbon molecules. So we will have 12 3PG molecules here. These 12 3PG molecules will then undergo some transformations to produce 12 of these 3-carbon molecules. Only 2 of these 12 3-carbon molecules will be able to exit the Calvin cycle, and the remaining 10 3-carbon molecules over here would go on to be regenerated to produce ribulose bisphosphate, our 5-carbon molecule. So ribulose bisphosphate is our 5-carbon molecule. We can see over here that there are 6 ribulose bisphosphate molecules regenerated because there are 10 3-carbon molecules used to regenerate this. So the 10 3-carbon molecules, 10 times 3, will give us 30 carbons in total, and our 6 ribulose bisphosphate molecules will give us 6 5-carbon molecules. So 6 times 5, in total, we will also have 30 carbons here. So essentially, the carbons here have just gone through a little rearrangement. So if we do a tally of our inputs and outputs, we already know that we have 6 carbon dioxide input to produce 1 molecule of glucose. So 2 of these 3-carbon molecules will combine to produce our 6-carbon molecule of glucose, C6H12O6. So 2 of these 3-carbon molecules combine to form a 6-carbon molecule known as glucose. So in 6 cycles of the Calvin cycle, we get production of 1 molecule of glucose. We also required 12 ATP here and 6 ATP here. This means that we used up 18 molecules of ATP in total. and 18 of these ATP molecules were converted into their low energy counterpart of ADP and their respective separate phosphate group. We also know that we required 12 NADPH to be used up in this stage, so 12 NADPH would therefore have also been converted to their low energy counterparts known as NADP+. Now, I also mentioned before that we get the production of 6 H2O molecules in this stage as well. These 6 H2O molecules can be considered to be generated from the oxygen that comes from the carbon dioxide and the hydrogens that come off the NADPH in this reaction to convert it into its low energy counterpart. The oxygens from the CO2 and the hydrogens from this NADPH reaction are combined to produce 6 molecules of H2O in this stage. So if we look at our unsimplified form of our photosynthesis equation, we can see that 6 H2O molecules are listed on the product side over here. Now, this form of the light-independent stage is what takes place in C3 plants. In our C4 and CAM plants, our first carbon fixation that occurs does not actually produce these 3 carbon molecules. Our very first carbon fixation that occurs within these plants produces 4 carbon molecules instead, and there are reasons for this, which we will discuss in just a second. For now, we should understand that in C3 plants, the very first carbon fixation that ever occurs in this plant is this addition of carbon dioxide to ribulose bisphosphate by Rubisco to produce these 3 carbon molecules called 3PG. Although we do have this 6 carbon intermediate, we generally do not write our equation in this way. We simply say that we get production of this 3 carbon 3PG molecule. So this 6 carbon intermediate is often overlooked. The reason that our C3 plants are called or named C3 plants is because the very first carbon fixation that occurs produces a 3 carbon molecule. So 3C, C3. So our C3 plants, our very first carbon fixation that ever occurs in this plant will result in the production of a 3 carbon molecule. This is different for C4 plants, in which their very first carbon fixation to a particular molecule will give us a 4 carbon molecule. This 4 carbon molecule will then be converted back into carbon dioxide, so that the second time carbon is fixated, it will be by Rubisco in this Calvin cycle. So the action of Rubisco in C4 plants is not the very first step of carbon fixation that occurs. The first carbon fixation that occurs will be to generate this 4 carbon molecule, which is why we call this a C4 plant. So 4C, 4 carbon molecules, so C4 plant. The second time after this carbon dioxide is released from this 4 carbon molecule, we do have Rubisco acting to begin our Calvin cycle over here, just as we have in C3 plants. But the very first time the carbon is joined to anything, it will generate a 4 carbon molecule. Okay, let's delve into this in a bit more detail. So our C3 plants initiate our light-independent stage in the most simple way. So as we discussed before, our C3 plants simply take in their carbon dioxide, and they take in the carbon dioxide through what we call stomata. Our stomata are simply pores found on the leaves of plants that regulate gas exchange and water loss. So when our stomata are open, this allows for gas exchange to occur, meaning that carbon dioxide can enter the plant and O2 can leave the plant. However, when these stomata are open, it is also possible to lose water through evaporation, or in the case of plants, this loss of water in the form of vapor is known as transpiration. So transpiration you can think of as plants sweating. So plants losing water in the form of water vapor. So when the stomata is open, we can get transpiration occurring. So the rate of transpiration is higher when the stomata are open. So in C3 plants, our stomata is open to get carbon dioxide to enter the cell. In C3 plants, this carbon dioxide will immediately be used by Rubisco to add it to ribulose bisphosphate and produce our 3 carbon molecule known as 3-phosphoglycerate. As mentioned before, the very first stable product that occurs due to carbon fixation is a 3 carbon molecule. This is why we name these plants C3 plants. Now, this is not ideal in warm and hot climates. This is because the stomata would have to remain open for a long period of time. This means that these plants would lose more water. Therefore, the pros of this plant can only be considered to be energetically efficient under cool or moist conditions. However, these plants would find it harder to survive in warm areas. Another disadvantage of these types of plants is that all rubisco molecules found in any plant, whether it be C3, C4, or CAM, they can also bind O2. So the rubisco can also bind O2 when our stomata is open. If rubisco binds O2, it allows for the process of photorespiration to occur. This is inefficient for photosynthesis because for photosynthesis to occur, we want rubisco to only bind carbon dioxide. So if the stomata is open and gas exchange of carbon dioxide and oxygen is occurring, we would only want carbon dioxide to bind to this rubisco. However, these plants have no mechanism to regulate which of the carbon dioxide or O2 will bind to rubisco. This would make them inefficient in areas where there is low carbon dioxide and high levels of oxygen. So these plants would probably find it harder to survive in environments where there are higher concentrations of O2 and lower concentrations of CO2. Now, our C4 plants have evolved to combat this photorespiration from occurring, and they've evolved to optimize rubisco binding to carbon dioxide to ensure that photosynthesis occurs. So in C4 plants, what they do is that they reduce photorespiration, or they reduce rubisco binding to O2 by separating the initial carbon fixation and the Calvin cycle. So over here, they spatially separate the very first carbon fixation that occurs and the carbon fixation that occurs in the Calvin cycle. So this is separated spatially. So to understand this, we must understand that the C4 plants have two types of plant cells within them. In our C4 plants, we have our mesophyll cells located on the outside and our bundle sheath cells on the inside. These mesophyll cells are in direct contact with the carbon dioxide and the oxygen in the air. So these mesophyll cells are able to choose between carbon dioxide and oxygen, and then they are able to stop oxygen from passing through these cells into the bundle sheath cells. So our mesophyll cells contain an enzyme known as PEP carboxylase. So maybe I should label this. So here are our mesophyll cells, and here are our bundle sheath cells. So within our mesophyll cells, so in our mesophyll, we contain an enzyme known as PEP carboxylase. As we know, our enzymes catalyze certain reactions. In this case, our PEP carboxylase catalyzes the reaction between carbon dioxide and a molecule known as PEP. This PEP is a 3-carbon molecule, and the addition of carbon dioxide to PEP produces a 4-carbon molecule known as oxaloacetate. This PEP carboxylase can only bind carbon dioxide. It cannot bind O2. This is why the mesophyll cells can ensure that only the carbon dioxide is able to pass through this layer of mesophyll cells and go to our bundle sheath cells where it meets rubisco for the Calvin cycle. So the reason that we name these plants C4 plants is because the first stable product of carbon fixation here is a 4-carbon molecule. Therefore, it is named a C4 plant. So this oxaloacetate can then undergo some processing to release a molecule of carbon dioxide from it, and this carbon dioxide is produced in our bundle sheath cells. So our oxaloacetate will undergo some processing to release the carbon dioxide in our bundle sheath cells. Now, if we notice over here, the bundle sheath cells are not in direct contact with the oxygen in the atmosphere. This means that the rubisco here cannot access the O2 molecules in the atmosphere. Instead, it can only receive the CO2 released by the transformations that occur to our oxaloacetate, which is generated by the activity of PEP carboxylase. So our bundle sheath cells and the rubisco in our bundle sheath cells cannot bind to oxygen, meaning that photorespiration will not occur, and it can only bind to carbon dioxide that is released from this 4-carbon molecule known as oxaloacetate. So once again, in our mesophyll cells, our PEP carboxylase, which only binds carbon dioxide and not oxygen, will join carbon dioxide to PEP to produce oxaloacetate. This oxaloacetate will then undergo some transformations to produce carbon dioxide, which can then be accessed by our bundle sheath cells. In our bundle sheath cells, this carbon dioxide will come across ribulose bisphosphate and rubisco, which will catalyze the production of our 3-carbon molecule known as 3-phosphoglycerate. So this molecule is the first of our Calvin cycle. So the rubisco over here will allow this carbon dioxide to enter the Calvin cycle. So the bundle sheath cells is where this carbon dioxide can enter the Calvin cycle. As mentioned before, we did say that there is a 6-carbon intermediate that forms before we get the production of our 3-carbon 3-phosphoglycerate molecules. But because this is a very unstable molecule, it is often overlooked. So we write our equation or our reaction that Rubisco catalyzes as ribulose bisphosphate plus CO2 producing our 3-carbon 3-phosphoglycerate. So it will produce 2 of these from that 6-carbon intermediate. Remembering that ribulose bisphosphate is a 5-carbon molecule. This carbon dioxide obviously has 1 carbon, so these will join to create a 6-carbon intermediate. However, this is unstable, so we don't often talk about it. And this will split to produce 2 3-carbon molecules. So it'll create 2 3-phosphoglycerates. Now, the pros of our C4 plants would be that they'd be more efficient in locations where there is less carbon dioxide and high levels of O2. A disadvantage of this process would be that the transport of this oxaloacetate to our bundle sheath cells to then release carbon dioxide would require more energy or more ATP to be used. So more ATP will be required in these plants for the transport of oxaloacetate to our bundle sheath cells, or the transport of carbon dioxide to our bundle sheath cells through oxaloacetate.
Note over here that while oxaloacetate is initially formed, it is usually converted to other organic compounds for transport. When this other organic compound reaches our bundle sheath cells, that is when carbon dioxide can be released from this compound in order to allow the carbon dioxide to enter our Calvin cycle.
Some examples of these C4 plants include things like corn or sugar canes. Okay, now last but not least, our CAM plants have evolved to survive in very, very hot climates. What these plants do is they temporarily separate the carbon dioxide uptake, so the first carbon dioxide fixation, and the carbon fixation that occurs in the Calvin cycle. So temporarily separating, for those who don't know, just means that we're separating these two processes in time. So this means that at nighttime, when the temperature is low, so as I said, these plants have evolved to survive in very hot climates. So at nighttime, these plants will open their stomata. This is more efficient than opening your stomata during the daytime when it's hotter, because if you open your stomata at nighttime, there's less sun, less heat, and so you will get less loss of water. So at nighttime, these plants keep their stomata open. This means that carbon dioxide can enter the plant, and this carbon dioxide is again used in a reaction catalyzed by PEP carboxylase, so the same enzyme used in our C4 plants. So we still have PEP over here, and PEP, our 3-carbon molecule, has carbon dioxide added to it, so that's 1 carbon, to produce a 4-carbon molecule known as oxaloacetate, which we discussed before. So this step is still the same. However, this oxaloacetate over here can undergo further transformations to turn this oxaloacetate into a different 4-carbon molecule known as malic acid. So this is still a 4-carbon molecule. This malic acid can then be stored within the plant's vacuoles. So this malic acid is stored within vacuoles. This means that during the daytime, we can keep our stomata closed because we already captured a lot of carbon dioxide during the nighttime. So we have no reason to keep our stomata open. So we keep them closed during the daytime, and this allows for us to minimize loss of water during the day when it is hotter than at night. So from this malic acid, we get the release of carbon dioxide, and this carbon dioxide can then be used by Rubisco during the daytime. Now, the reason that Rubisco acts during the daytime is because during the daytime we have sunlight, meaning that the light-dependent stage of photosynthesis can occur and generate ATP and NADPH required for our Calvin cycle. So our carbon dioxide can only enter the Calvin cycle when we have the ATP and the NADPH ready for it to occur. So this carbon dioxide during the daytime is released by malic acid. So this carbon dioxide is released by malic acid during the daytime, and Rubisco can catalyze its entrance into the Calvin cycle by adding it to ribulose bisphosphate to generate our 3-carbon molecule known as 3-phosphoglycerate, so just as before. So the carbon dioxide can now enter the Calvin cycle due to the action of Rubisco during the daytime. So as mentioned before, these plants have evolved to survive in very high temperatures. So some examples of these cam plants would be those that I find in deserts. So this could include plants such as cacti. So if we summarize our C3, C4 and our cam plants, it is important to know that the first product of carbon fixation in these plants is different. In our C3 plants, our first product of carbon fixation is 3-phosphoglycerate, whereas in our C4 plants, the first product of carbon fixation is oxaloacetate. And in our cam plants, our first stable product of carbon dioxide fixation is our malic acid. So this is only at nighttime though. The enzyme responsible for this initial fixation is also different in these three plants. So the enzyme responsible here would be rubisco. Over here, we would have PEP carboxylase. And over here, we would also have PEP carboxylase, but once again, only at nighttime. Our location of our Calvin cycle is also different. In our C3 plants, our Calvin cycle occurs in our mesophyll cells. In our C4 cells, this occurs in our bundle sheath cells. And in our cam plants, the Calvin cycle occurs in our mesophyll cells, but again, this happens during the day. So only during the day. Alrighty. Hopefully, this gives you a bit of an overview of photosynthesis, and learning a little bit about the light-dependent and light-independent processes will help you remember the inputs and outputs of each stage of photosynthesis. As always, let me know if you have any questions. Otherwise, I will see you next time.